Welcome to Recovering Evangelicals, a podcast for people who were once very comfortable in their Christian faith until the 21st century intruded and made it very hard to keep on believing. I'm your host, Luke Jansen.
And I'm Scott Dyer.
Scott, good to get together with you. Happy Thanksgiving weekend to you.
Thank you. Yeah, no, we're looking forward to it.
For the listeners, I should say, this is Canadian Thanksgiving weekend. If you're from America or elsewhere, it's not. Yours is coming, but for us, it's Thanksgiving. Are you guys doing anything, Scott?
We're going to a friend's for some turkey, or I assume turkey. I don't really know, but it's just going to be a quiet Thanksgiving weekend.
Okay, well, Scott, I've got a plan for you. I want to take you on a camping trip out in the deep woods of northern Canada for a month or two. Are you up for it?
I don't know about a month or two. I'd love to camp. Depends on what kind of equipment we have.
Well, that's exactly where I'm going with this. So here's the details. You've done a fair bit of camping, I'm sure. What are some of the things we need to bring for a trip like that? Again, 2 months in the remote woods up north.
Yeah, interesting. I've been watching this new show called Alone, where that's what they do. They drop people in the woods with, just whatever they chose to bring, which is very, have to give them 12 items or something. So, I guess something to start a fire would be probably my number one thing, and something to chop and kill things with, if you're talking about survival.
Right?
Yeah.
Well, you can bring more than 12 things, so you're going to want a tent, you're going to want a sleeping bag. Just name a few more things.
Yeah, now we're getting luxurious. Okay, sure. A tent, backpack, some sort of cooking apparatus, a water purifier. I'm trying to think, because I've done some pretty serious backpacking in my day.
How about food?
Oh, well, it's a good point. That's a good point.
That's a good point. Yes, exactly.
I'm thinking of foraging. Sure. Pack some, especially dry food, that we can rehydrate. Because you said the deep woods, so, I'm assuming that we're going to want to go light.
Right.
So probably a lot of freeze-dried stuff. And maybe for the first night, something a little nicer, because we're going to eat it right away kind of thing. Sure. Maybe some frozen chicken or something like that. Yeah.
Okay. So that's a pretty good list, a lot of stuff. But let's change the scenario up a little bit. Let's just say I won free tickets for a 2-month stay at a hunting resort, a sprawling lodge in the middle of nowhere with all-you-can-eat buffet. Would that change how we get ready?
Yes, significantly.
How so?
Change our pocketbook too. Obviously wouldn't need to bring any food or food-related equipment. It would change everything, I would think.
Just leave the food behind, forget the stove, forget the sleeping bag.
Yeah, some warm clothes and appetite, I guess. And my hunting rifle. I don't have one. I don't hunt, but anyway,
Okay, so now, another change of plans. Let's just say we get up there, and 1 week in, the camp director announces that the refrigerators went on the fritz, and a lot of the food spoiled and had to be pitched. There's enough food for 4 or 5 days, but the next shipment of food will take another 2 weeks to get there. So the camp director tells us to limit the daily amount of food coming out of the kitchen down to 1/4, but it's still going to be an all-you-can-eat buffet. What should we do?
Well, how can it be an all-you-can-eat buffet if he's rationing it down to 1/4?
Well, that's the question. you get in there and eat another man's breakfast. It's basically, literally, it's actually a situation of you snooze, you lose, right?
Yeah, right. Yeah.
Okay.
Yeah, I mean, the tendency in a situation like that is you probably get very selfish, right? You probably start looking at your own survival and not caring too much about sharing.
Right. Okay. Now, one last development in this story, and then I'll get to the point.
Yeah.
An earthquake happens. The camp is destroyed. The devastation makes it impossible for planes to come. In fact, the bush and the mountains make it impossible for helicopters to get us. So there's a town 100 miles south of the lodge. We can hike it back about 4 days through thick brush. So what do we bring? What do we do?
We salvage what we can to carry on the trip. But we head out of the camp.
Okay, and probably break some furniture first because we have to make snowshoes or skis.
Yeah, okay. I didn't say it was winter. Okay, sure.
Yeah, tear up some of the linen to make knapsacks, right?
Yeah. So in other words, we would convert what we could into survival type stuff, right?
Right.
Yeah. Yep.
Okay. Where am I going with this? That was an exercise to get us thinking about survival, what we need to do to survive, and how That can change radically when all our daily needs are taken care of for us, and then how that all changes when we have to compete for limited resources. And tough decisions have to be made, like taking resources from the guy beside us, breaking a few things, repurposing things, getting resourceful. And that's the story of evolution. Life, living organisms having had to adjust to changes in their environment, coming up with new innovations, new solutions to new problems, and in the process becoming new species.
Yeah, the driver there, like you said, it's survival. That's the overwhelming driving force. It seems to be in the natural world, is that some way or another, organisms. I remember the movie Jurassic Park, the first movie, and you probably remember the line, "Life will find a way."
Right.
Right.
Jeff Goldberg.
Yeah, exactly. And of course, that is kind of anthropomorphic, because life doesn't find a way. It's just that given we're in a harsh environment, life's drive to propagate will cause it to adapt or die, kind of thing, right? Yeah.
Exactly. Over the past 2 months, we've been taking a deep dive into intelligent design, even speaking directly to two leading proponents of the ID worldview. And it goes without saying that evolution theory came up frequently in those discussions. A lot of talk about the genetic mechanisms behind evolution. A few times, references were made to Richard Lenski's work, growing E. coli bacteria up through thousands of generations, a long-term evolution experiment which later became widely known as, well, the long-term evolution experiment, or the LTEE. And in the same way that ID proponents speak often and incorrectly about, okay, trigger alert here, some people like me now exhibit a neurotic twitch whenever we hear this next word, the bacterial flagellum. That word, that topic may be the most overused talking point employed by the ID advocates, but the second most common one is probably the LTEE, and maybe junk DNA is a close rival. And as expected, because they do this all the time, just like they completely misrepresent the whole flagellum story, they also completely misrepresent the Lenski experiments, the LTEE, by always claiming that the only thing Lenski's group showed was how to break genes. To me, this is no different than Trumpers always referring to the stolen election. It's about time that they just give that up. And Scott and I can say that they are misrepresenting the true story because we talked to the Lenski group directly. Too many other people, like these creationists, try to become their own experts. They think they're competent to just read the scientific papers and come up with interpretations that are quite different from, sometimes even opposite to, the interpretation of the people who have been engaged with the question and the work for decades. The ones who have actually done the experiments and actually done the complicated analysis.
Yeah, for sure. We actually talked about that kind of arrogance and kind of craziness last week with Dr. Janet Kellogg-Ray, when we were talking about science denial and pseudoscience and people thinking that they can be their own experts. And of course, with the proliferation of knowledge now vis-a-vis the internet and now the rise of artificial intelligence and so on, this is even going to become exponentially, I think, a greater issue with the idea that, we can kind of be our own experts and substitute our supposed expert opinion for real expert opinions simply because they're not comporting with what we want to hear,
So here's part of an interview I did with one of the members of the Lenski group, one of the many people who actually did the work and the analysis and who has since started his own genetic research lab, which is branching off from and building on the original Lenski experiments. One of many people from the original Lenski group to do so. Which, by the way, that's a healthy sign, a hallmark of great science when it leads to new ideas, new questions, and new research directions. And our guest he was a very engaging guy to talk with. I'll introduce him in a few moments, but first I want to give the listeners a bit of background information and a framework to hang these ideas on. I did that in our earlier episode when we talked to 3 world-leading biologists about how an ancient bacterial nanomachine evolved over billions of years into half a dozen other nanomachines, including, again, trigger alert, the bacterial flagellum.
It's just funny. I have a great interest in microorganisms, and I just for fun, I will often collect specimens and just observe them through my microscope, just find them fascinating, even though I don't know a lot about them. So every time I see one now with a bacterial, it won't be a bacterial flagellum, but with a flagellum,
Yep.
I'm going to feel bad.
Right. Okay. So our guest will explain all of this in greater detail, but again, I want to put this starting image or framework in the minds of the listeners. Richard Lenski started the long-term evolution experiment at UC Irvine 40 years ago, and then more recently he moved it all to Michigan State University, and then just last year he handed off to a team at the University of Texas at Austin. We'll hear more about that, the history from our guest. In this experiment, Richard Lenski simply started growing bacteria for generation after generation, and they're currently up to 76,000 generations, which, if you put that into human terms, where a human generation is 15 or 20 years, that equates to about 1.5 million years. And that sounds like a lot, but in evolutionary terms, it's barely a blink of the eye. I mean, the first mammal arrived on the world stage 250 million years ago. And that world stage was already populated by reptiles, birds, and fish and insects and plants and bacteria. And humans diverged from chimps 6 million years ago. So 1.5 million years is still not a very long time, but just the same, Lenski's team have already identified a long list of dozens of genetic changes that have occurred in that short period of time.
Interesting.
They've been freezing samples of the bacteria every 500 generations, which they call their frozen fossil record. I thought that was a really insightful thing that they did.
Yeah.
And they did that so they can do deep dive experiments and as well recover from unexpected problems. And here are a couple really important details to put on your mental framework of what's going on in these experiments. Our guest will refer often to the two main ingredients that cells need for survival, to survive and reproduce, and those two things being oxygen and fuel. So he'll talk a lot about aerobic conditions, which simply means in the presence of oxygen, and anaerobic conditions, the absence of oxygen, like when those bacteria are swimming around in the fermented poop of your intestines. This is why the bugs are called E. coli. They live in your colon, your intestine. The coli part of that name literally means of the colon.
That's interesting.
The listener may have also encountered this term anaerobic metabolism when they've heard about fermentation, the very slow and controlled burning of sugars, kind of like putting a pan on the stove that's boiling over, the equivalent of aerobic metabolism, turning that down to a low simmer, the equivalent of anaerobic metabolism.
Yeah, we make kombucha as well, and that's another fermentation process that takes advantage of some of those processes, anaerobic and anaerobic. I also used to be quite an avid keeper of aquariums, especially marine aquariums, and anaerobic bacteria were some ways kind of the enemy, because what the problem in an aquarium, they give off nasty gases. I forget the one
Methane?
in particular now. Yeah, I think it was methane. Which is, of course, now in small quantities. The good news was that could be helpful, but if you get large quantities of those bacteria, then of course it'll kill the fish. So yeah.
So that's the oxygen side of the story. I said there was two. He also talks a lot about fuel sources, and he'll really only refer to a few of them. There are, in fact, dozens or hundreds of different bacterial fuel sources. Basically, all the simple molecules that come out of the food that your stomach and stomach acids have worked on. So things like sugars, amino acids, carbohydrate acids, and fats. So our guests will often refer to glucose, a simple sugar, and once or twice to ribose, another simple sugar, and often to a related carbohydrate acid called citrate or citric acid, the one that you taste in oranges and other citrus fruits. And he'll get into detail about citrate metabolism, and he does a great job explaining that, so I'll leave that up to him. But I just want to make sure the listener understands that the Lenski group never expected this window on evolution, the whole connection to citrate. That was a complete surprise to them, and a very powerful one. One of those things, the kinds of things that make doing scientific research so much fun to do.
And maybe as somewhat of an analogy, our listeners might be familiar with the whole keto fad, I won't call it a fad, but the whole keto diet emphasis these days. And that's kind of a classic example, because the idea there is you starve your body of sugar at it. And then instead of your cells burning simple sugar, which can often lead to fat. It burns another substitute kind of for sugar, and that actually leads to weight loss and so on. So it's kind of an interesting example of how we can use our own bodies as a
Yeah,
to show what happens when our bodies can't get what they would normally
exactly.
want, and
Yep.
then you actually switch and start burning something else. So yeah.
Okay, Scott, so now it's time to bring in our guest. Unfortunately, Scott couldn't be there with me for this interview, but he was kind enough to let me go ahead without him. So now we're going to play the first half of the interview I had with our guest, and then Scott and I are going to talk a little bit about what he said. So, Scott, grab a coffee and come back in 45 minutes.
Okay, I will.
Our guest today is one of the people at the front edge of the Lenski team we've been talking about. Dr. Zachary Blount earned a Bachelor of Science in Applied Biology at Georgia Tech, and then did graduate work at the University of Cincinnati, looking at the genetics of a certain specialized strain of ancient bugs. Not bacteria, but archaea. Again, as we mentioned a couple weeks ago, bacteria and archaea were the earliest forms of cellular life on the planet. And the bugs that Dr. Blount was looking at are referred to as thermoacidophilic, meaning they live and thrive in the boiling acidic hot springs that you might find in Yellowstone National Park and other volcanic places around the world. And then Dr. Blount, or Zachary if I may, joined the long-term evolution experiment team led by Richard Lenski at the University of Michigan, and Zach is now research assistant professor and director of graduate education innovation in the Department of Microbiology and Molecular Genetics at MSU, and he's also an honorary senior research fellow in the Department of European and International Social and Political Studies. So, Zachary, welcome to the podcast.
Hello, it's nice to be here, and please call me Zach. That's what everyone else seems to.
Zachary, why don't we start with a bit of a history of the LTEE, some of the key players, and what were the basic goals of the experiment, and most importantly, how did you fit into this story?
Goodness, it's quite a long story at this point. Basically, it comes down to, it was started by Rich Lenski in 1988, and Rich started out as a field ecologist. He studied beetles in the mountains of North Carolina, and he wanted to start testing hypotheses experimentally. But it's really hard to do experiments in the field. You know, thunderstorms would destroy apparatus. He had to deal with venomous snakes and so on. And he started wondering if there was a better way. And he remembered work that he had really appreciated learning about in college and grad school done with bacteria, especially some work done in the 40s by S.E. Luria and Max Delbruck that demonstrated the randomness of mutations using bacteria. And he realized, well, bacteria could really be used to address these evolutionary and ecological questions that I'm interested in. And he ended up going to work with Bruce Levin, who at that time was at University of Massachusetts Amherst and is now at Emory University in Atlanta. After he learned how to do microbiology with Bruce, he got a position at UC Irvine. And he kept thinking about how he could address some very fundamental questions in evolution using bacteria. And one of the other professors at Irvine was Michael Rose, who had been doing an evolution experiment with Drosophila to see if he could extend the lifespan of Drosophila. And Rich thought, well, why don't we do an evolution experiment with E. coli? And so in 1988, on February 24th, he took 12 colonies of a clone of E. coli called REL606, and he used each of those colonies, which had been founded from a single cell, to inoculate and start 12 populations that initially were identical. And ever since then, every day, someone has come into the lab, taken these 12 populations out of the incubator, and using a micropipette, transferred 1% of each population to a fresh volume of the growth medium, and then they're put back in the incubator. And it's just been...
And
yes, go ahead.
And that was something that was done every day. And if I'm not mistaken, that represents a few generations. So every day you're transferring a few generations.
Well, every day, because we're transferring 1% of each population. So every day the populations grow from 1% to 100%. So there's a 100-fold increase in the population size. That corresponds to about 6.67 generations every day in each of the 12 populations. And by every day, there's some wobble there because there have been times when we've had to go back to restart populations from frozen stocks because we freeze the populations every 500 generations as a backup and also as a research resource. And then sometimes the populations have had to be put on ice for when, example, Rich moved from Irvine to MSU or when we had to shut down for COVID. So it hasn't been literally every day, but under ordinary circumstances, it's every day someone comes in and does the simple procedure of transferring a small volume of fluid from each flask containing a population to a flask of fresh medium. And that has been going on now since the Reagan administration. We have a lot of frozen samples from different time points in history. Dozens and dozens, if not hundreds of people have worked on the experiment. Rich just handed the experiment off last summer to Jeff Barrick at UT Austin, like you mentioned. Jeff is a former postdoc in the lab, incredibly brilliant individual, very kind person too. And under his direction, the experiment's going to continue for a few decades, and then he'll hand it off to someone else so that we can keep this experiment going far into the future to help us provide resources to continue to plumb the unknowns of evolution. Now,
and what about you?
Yes, I was about to get to myself. So I came in, I would say, at about generation 35,000 in 2004. So I started at MSU in the PhD program in microbiology and molecular genetics in 2003. And the first year was rotations through different labs. And I initially thought I was going to work on astrobiology with James Teege, but my final rotation was in the Linsky lab. And I just fell in love with it. It was such a wonderful system. The people were kind. The science being done was spectacular. And I started working with this population that we'll talk about later that evolved the capacity to grow on citrate aerobically, which E. coli is not supposed to be able to do. And I've stayed ever since, with the exception of a year I spent teaching at Kenyon College, studying all sorts of phenomena, because it turns out this one population has yielded a huge number of questions and phenomena that provide for a lot of very interesting research work.
And before we get into the actual data, I want to expand on a few things that you just said. First of all, you just finished saying something a minute ago, something about citrate. A lot of people aren't going to know what that's all about. Why do you specifically add citrate? Why does the cell normally not do anything with it? Just tell us a bit about that.
Right. So citrate basically, that it's an acid. It's what makes oranges and lemons tart. So many of the listeners will have likely eaten some of it today. It's something that E. coli cannot grow on aerobically. It doesn't synthesize.
Meaning in the presence of oxygen.
Exactly. With oxygen there, it isn't able to synthesize a transporter to bring into the cell. So a transporter is like a mouth for a given substance. It's unable to synthesize a transporter, can't bring the citrate into the cell when oxygen is present, though it is able to, when oxygen is not present, to bring the citrate into the cell to ferment it into other substances, especially another acid called succinate.
So the cell uses oxygen to burn up glucose and get energy from that. But when oxygen is not around, the cell can resort to fermenting the glucose, anaerobic metabolism. But that doesn't return as much energy, and the cell looks for other ways to get energy in addition to fermenting the glucose. Fortunately, under those anaerobic conditions, the cell becomes able to make a transporter for a whole different kind of fuel, citrate, which can also be metabolized anaerobically, although that returns even less energy than the glucose under the same conditions. It's kind of like making a campfire using nice, hefty pieces of oak that have been drying for a year or two, versus scouring the woods for thin green branches and leaves from the ground. The oak logs are going to make a much warmer fire than the branches and leaves and burn a lot longer, and will all give even less energy back if they're a little bit damp, which is analogous to burning glucose or citrate anaerobically, but you'll do whatever it takes if you're literally freezing to death.
Now, as far as why it's in the medium if E. coli doesn't use it, that's actually a very funny story. I researched this when I was doing my dissertation, and it actually doesn't make a whole lot of sense. There's this massive quantity of citrate that's in this minimal medium that we provide the E. coli with just a tiny amount of glucose as food. I was puzzled, like, why is that there? I mean, we always say it's there to help the bacteria acquire iron, and that's true. But why is there so much? And it turns out that prior to knowing about that role for citrate in iron acquisition in E. coli, back in the 1950s, there was a professor at Cornell named Bernard Davis, and he was developing a method for isolating mutant E. coli that would be unable to grow on certain substances. And he used a selection protocol that uses penicillin as a selective agent. And because penicillin only kills actively growing cells. So if you put E. coli into a medium and you want to find mutants that are unable to use a resource, you provide that resource as the only thing they can grow on. And you have penicillin there. The penicillin kills anything that's growing, so anything that isn't growing can't use that substance. And he found that if he added citrate, it helped the penicillin kill better. And so he thought, well, okay, citrate is something that E. coli is inert toward. I don't have to adjust the concentration precisely, so I'll just add half a gram per liter and call it a day. And that helped the penicillin kill, we know now, because it helped the bacteria acquire iron and grow better.
That may have been hard to follow. Don't worry about the penicillin part of that story. The important thing to know here is that this professor, who was studying E. coli, just found that his experiments worked better when citrate was in the medium. Years later, other scientists found out that it had to do with citrate helping the bugs absorb iron, which the cell needs to be strong. Remember what your mom told you about eating your spinach. But this guy didn't know that, but just knew that it worked better when citrate was around, so he modified the lab recipe for growing E. coli to now include a few handfuls of citrate.
And then no one changed the formula.
Okay.
And yeah, there's kind of an inertia to laboratory science. No one saw any reason to change the recipe of that medium. That was the medium that was being used in Bruce Levin's lab. And when Rich started the long-term experiment, that was the medium he used, the citrate included. Now, he did try to remove the citrate to simplify things as much as he could. And what he found was that the growth was inconsistent. And it ultimately seems to depend on how much iron was in the water used to make the medium that day. And sometimes the bacteria would grow without citrate, sometimes they would not. So he decided just to include the citrate and thought, okay, maybe it's possible someday they'll figure out how to grow in this, but I don't really care. And after the first couple of years and none of them evolved the ability to grow in this citrate, then he just assumed they're never going to do it. Until early 2003, when someone came into the lab to do the transfers for the day and found that one of the populations was much cloudier than the others.
Which means
that there were more bacteria in that population than the others. That the bacteria were growing on something other than the glucose. And so Tim Cooper, that was the postdoc's name who found this, let Rich know. They plated it out, and it was full of a lot more bacteria than it should. And they looked like E. coli. They smelled like E. coli. And Rich thought, we'll follow the lab rule. When in doubt, throw it out. So they stopped that population, went back to the freezer, and restarted it from the last save point.
Meaning the last time they put a bunch of cells in the freezer. Remember, every 500 generations, they froze a bunch of their cells for future use, in case they wanted to do some specialized evolutionary research, or, in this case, if there was a technical problem and they needed to start over, without going all the way back to step number 1. They refer a few times to these frozen samples as their frozen fossil record.
And then a few weeks later, it got turbid again. It got cloudier. The bacteria grew to a higher population size, at which point he decided to think, okay, we should look more closely at this. And it turned out that the bacteria in the population had evolved the ability to grow in the citrate many years after the start of the experiment. And that ended up setting up my coming into the lab the next year to start really looking at aspects of the evolution of this novel trait.
Okay, so let's get the audience caught up then. basically, we're talking about citrate because it's going to become a very important part of the story. But
Right.
initially, citrate was just one of the ingredients that your team didn't think was important. You found you needed to keep it in, but you just didn't know why, so you kept it in. And that's where the story begins. I just wanted to emphasize then that the cells already, even from the very beginning, had the ability to take citrate up. They didn't in the presence of oxygen. It was able to use the citrate that it took up when there was no oxygen around. It could use it as an energy source, right?
Yes.
But otherwise, it didn't bother taking up the citrate because it just took too much energy to do so. And then the story begins to change. This cloudy bacterial solution that Tim found.
Yeah. Just let me go back. It's unclear exactly why E. coli normally can't grow aerobically on citrate, because there are closely related bacteria to E. coli like Salmonella that are able to grow aerobically on citrate. But for some reason, E. coli lost the ability millions of years ago. And it's possible that it's just that E. coli doesn't encounter citrate in its natural environment very often when oxygen is present. So there was no selection to maintain the genetic basis of being able to grow aerobically on it.
Okay. And it's not that the citrate uptaker itself was broken in any way, but the promoter, the on switch in the gene wasn't present or wasn't functional.
No, the so E. coli has an operon called the CYT operon, CYT for citrate. An operon is a group of genes that are turned on and off together. And the genes in the CYT operon control fermentation or anaerobic without oxygen growth on citrate. And our E. coli, the ancestor, had this operon. And it is under the control of a promoter that only turns the genes on in the presence of citrate but absence of oxygen. Needless to say, we conduct this experiment under atmospheric conditions because it's comfortable for us who are oxygen breathers.
Right.
And because oxygen was there, this capacity to ferment citrate was just always turned off. And we found out later there is an insertion sequence, a kind of jumping gene that is inserted into the promoter region controlling this operon. But, insertion sequences can move. So presumably if we put E. coli from our experiment under conditions without oxygen and gave them citrate to grow on, in due course, we would expect variants to arise in which the insertion sequence had moved on, the promoter would be active again, and they would be able to start growing on it.
Okay. So we're getting into quite a bit of genetics there. Maybe we should hold off for the moment. And let me just then talk again some more about the design of this experiment. Yes. You began growing these bugs, transferring them on a daily basis, growing them through thousands of generations, and you were just trying to see how they might evolve. You weren't thinking anything about citrate, and then we'll just set citrate aside for the moment. But I'm assuming you had some kind of a stress on this population because you were looking for changes. I think usually genetic changes they can happen randomly, and we'll talk in a minute about what random might mean or not mean. But otherwise, you biologists, you geneticists will try to move the timeline along more quickly by introducing some kind of a stress, competition for a resource. So can you tell us more about your experiment, how that was designed in?
Certainly. So Rich started the long-term experiment with three original goals in mind, three broad goals. The first was to look at the general pace of evolution. So does the pace of adaptation change over time? Does it slow down? Does it stop? Then he wanted to look at the relationship between change in phenotype, so the expression of the genes that the organism has, and change in the genotype.
Phenotype comes from the genotype. Phenotype refers to the results of gene expression, the proteins or functions that you can see or measure. So, for example, certain genes control hair and eye color. Being blonde and blue-eyed is the phenotype, and having genes XYZ and DEF are the genotype.
So looking at changes in the organism and change in the DNA and how that corresponded. That was a bit of an advanced idea at the time because we didn't have good sequencing technology. He figured that, the technology would catch up. And then third was to look at the repeatability of evolution, because this was an experiment in which you started from genetically identical cells in all 12 populations. All 12 populations were identical at the beginning. And they were evolved and have been evolving and are still evolving under identical environments that we provide to them. So if evolution is highly repeatable, you'd expect that all the populations would evolve the same way. But if it's possible for evolution to go in different directions, you would see the populations do different things. There's that. Now, as far as stressors go, I get this question, and I still have a bit of a hard time answering it because we don't really impose what we think of as a stressor, like an antibiotic or a selective agent. We provide the environment. We provide this minimal medium that contains a small amount of glucose as food. And it's very different overall, this environment, from where E. coli is normally found, which are the guts of vertebrates. So you could say the entire environment is stressful in a sense. But the primary thing that natural selection seems to be acting on is competition for glucose. And so the populations have adapted to these conditions that we give them, with a lot of the evolution having to do with improving the ability to make use of the glucose in the medium under the conditions we give them.
Okay. So that's interesting. Like, you weren't trying to nudge them into mutating. You were just trying to see when and how they would just decide. I'll use that word in air quotes, just decide to mutate. They weren't pushed in a certain direction.
Well, the mutation was going to happen anyway. It was a matter of they would adapt, but we didn't know how they would adapt.
Right, exactly.
I say we as though I were there with Rich in 1988 when he did this. Basically, the idea was give the bacteria an environment, keep giving them that environment, and just see where natural selection goes.
Okay.
And there wasn't any preconceived notion of we will evolve bacteria that are able to do this. Instead, it's just a matter of provide the environment, see how evolution occurs, how it goes.
Okay. So a few minutes ago, you're telling us about the objectives, one of which was repeatability. You wanted to look at that. And yet now we're talking about mutations and the fact that they might be random, which is not repeatable or not predictable. So maybe tell us a bit about this randomness, in air quotes, the randomness of mutations that produce the genetic variation.
Right. So modern evolutionary biology, which is based in large part on the idea that adaptation only occurs by natural selection. And natural selection, this principle that the environment selects among randomly generated heritable variation. And whatever gets selected gets passed on, and then the cycle repeats over and over again with changes, accumulating the organisms becoming more and more adapted to whatever environment they're living in. Now, the notion of random variation was something that was held to be the case for quite a while without having proof positive that it was in fact random. And there were, of course, people who came after Darwin who rejected natural selection specifically because they couldn't cope with the idea of randomness being involved. So you had, during this time called the eclipse of Darwinism, people accepting evolution, but insisting that it had to work in some way other than natural selection so that it wasn't based on random variation. And so there were theories like theistic evolution, where God was guiding it all. This was actually part of science until the people who were behind it realized this isn't really science, and that lineage was rejected. The neo-Lamarckism, a resurgence of the idea that acquired characteristics can be passed on, or orthogenesis, that there is some force that causes organisms to evolve. That, of course, was rejected in the early 20th century once it began to be realized that natural selection really does work once you combine it with Mendel ideas about genetics. So by random variation, we don't mean uncaused changes. We now know, of course, that variation is introduced by changes in DNA, by mutation, recombination, and so on. And those changes that generate this variation are random. So the variation that's generated is random. And when we say random, we don't mean uncaused. We mean that it is random with regard to the selection acting on the population. So that if organisms are exposed to, say, antibiotics, there is not a bias to produce variation that specifically provides resistance to the antibiotics. It's just random, and then selection may select variants that where mutations provide resistance to antibiotics. Now, this was demonstrated in the 1940s by the two individuals I mentioned earlier, S.E. Luria and Max Delbruck. And they were at the University of Indiana, and they were thinking through this question that had been had for a while, which is bacteria seem to evolve not in Darwinian ways, but in Lamarckian ways. Because if you expose bacteria to any selected agent, like a virus, a phage, they very rapidly evolved to counter the phage, to be resistant to it. And so was this notion that, well, bacteria, they just have variation that arises in response to a selective agent. And they were wondering, how can we test this to see whether or not these mutations are actually random or if they are in response to selection? And there's the story that Luria told that he was at a faculty dance and there was a slot machine and he was watching people play the slot machine and he saw jackpot. And that gave him an idea that, okay, if a mutation occurs very early on in a population's growth, then all of its descendants will have that mutation. So that insight led him and Delbruck to do this really amazing experiment called the fluctuation test, in which they grew replicate populations of E. coli from very small numbers of cells to ensure that none of them had the same mutation that had arisen since the ancestor. He grew these populations up and then they plated them on petri dishes containing a virus. And what they realized was if mutations occur in response to selection, then they should see the same number of mutant colonies arising from mutant cells that were resistant to the virus on all of the plates. Whereas if mutation was random and occurred prior to selection, then in some of those populations, the mutation that provided resistance to the phage would have occurred early on, in which case there would be a jackpot. And a huge number of the cells that got on the plate were resistant, whereas some, it would occur right before plating and you would only see one or two colonies. And that's exactly the pattern that some plates had none, some had one, some had thousands. That was the first demonstration ever that mutations were in fact random and that selection acts on randomly generated variation instead of variation that's specifically created to counter the selective agent.
Okay.
Yeah.
And just one more clarification on this whole topic of mutation, because of the fact that creationists and ID proponents I've found, they talk about mutations as being a point mutation, just individual base changes. But the mutations can also include movements of large chunks of DNA. They can move around, even sequences within the gene. The gene itself can be sometimes in a way shuffled. So my point being, it's not just simply individual base points, but even large sections of DNA.
Oh, absolutely. Absolutely. Right. Yeah. When we say mutation, we generally mean a range of different changes to DNA. So they can be point mutations that affect individual base pairs. They can be insertions of either insertion sequences, these jumping genes, or even of segments of DNA that are from other places in the genome. You can have inversions where the DNA just kind of flips around. You can have large deletions. And then, of course, you also have extra variation created by recombination between different cells or between areas of the genome of a single organism. Right. Yeah. So it's not just point mutations. No.
Exactly. Right. Okay. So then let's move away, zoom out from the whole topic of genes and mutations. And let's talk about this experiment. Your team are growing these bugs on a daily basis, passaging them on, and you began to see some changes. So what did you guys find?
As you might imagine, it's kind of difficult to summarize going on 40 years worth of work.
Exactly.
And I think at this point, 300 or 400 papers that have been done in the experiment.
Wow.
Yeah, it's remarkable. humbling, even if you're a part of it. It's like I look at colleagues and I think, wow, you've done such amazing work, and I only vaguely understand it because it's so far removed from what the portion of the experiment that I
And
studied.
you only vaguely understand it. What are we going to do?
Uh, hope that I explain it well enough that it is at least vaguely understood by the audience.
Okay. Okay.
So we've seen a number of things. For one, as far as repeatability goes, we've seen a mixture. So over the course of the experiment, the populations have all improved in fitness in this environment we provide them. So the bacteria from the populations now around 76,000 generations, they grow about twice as fast as the ancestor did. And if you look at their fitness trajectories, so the change in fitness, there was a rapid improvement in fitness very early on, and then it slowed down. This is true for all of the populations. They have the same general pattern, though their trajectories are subtly different. This indicates that they're doing much the same thing evolutionarily, but not quite. And if you drill down and you look at the genetic level, you'll see that there are a number of genes that have been mutated in a number of the populations, and in some cases, all 12. So these are adaptive mutations that are being found in parallel across the 12 populations. But not all of them have the same adaptations. Each has its own unique suite of mutations. So they're no longer genetically identical. They're doing some of the same things to adapt, or they've adapted in similar ways, but not exactly the same. Other things we've noticed is that all the populations have cells that are much larger than the ancestor, which was a surprise. There was actually an expectation that the cells would become smaller over time. And even though they've gotten larger across all 12 populations, they're not the same size across all 12, and the cell shapes are not the same. So again, similar directions, but not identical. Going back to the fitness trajectories, this is really nifty. This is something that has come out of the experiment that no one really suspected. There was this notion that in a given environment, if it held stable, natural selection would drive adaptation to a certain point at which the populations were as adapted as they were going to be. And at that point, what selection would do, it would purify. So it would keep out detrimental mutations, but keep it up at the top of that fitness peak, we call it. So that would suggest that after a time, the populations just stop improving. But what we found is that's not what happened. The populations are still getting better, just more and more slowly. And we have evidence from first principles, from theory, and theoretical models that can recapitulate what we see with these fitness trajectories that indicate that the populations will still be improving in their fitness hundreds of thousands of years from now, just more and more slowly.
Okay.
So you can't actually get to the top of the fitness peak just because eventually you just have a lot of mutations of very tiny effect, and it takes them a very long time to get selected for and go to high frequency population.
Okay.
Yeah, no one knew about that. So this experiment has yielded a lot of nifty results and continues to all.
And so given that the different cells took different approaches to solve similar problems, that goes against the whole idea that any of these things were pre-programmed changes, pre-programmed responses to an anticipated stress of any kind?
Right, I would say so, because you would kind of expect if there was a programmed response that you would see parallelism all the way down to the mutational level. And when I said that you see many of the same genes mutated in all 12 populations, it's very rare that it's the same mutation that you find in each population that has this gene mutated. So that says that mutation is leading to different variants in which there are mutations in these same genes across populations. And just when it occurs in one population, it gets selected for. If a different mutation occurs in that gene in another population, it gets selected for. So it's very much speaking to the randomness that's generating the variation that gets selected.
Okay, so Zach, this is a fascinating. You said 300 or 400 papers. A lot has been learned. You gave us just a thumbnail sketch of what some of the things that have been found. Can you give us some specific examples, maybe, of some genes that had been changed? When Zach mentions each of these, he'll give the name of some specific gene that they found had changed, that were different in the more recent populations compared to the original ancestor population of E. coli that they started with. And they proved that it was those specific mutations alone that were beneficial by specifically inducing the exact same mutations in the genes of otherwise healthy, non-mutated control cells, and showing that there was a measurable increase in fitness, in growth, often measured in terms of percent.
There are maybe 57 genes, at least by, I think, 70,000 generations that have been mutated in parallel across the populations. And, some of them really stand out. There's one that occurred very early on where in every one of the populations there was a deletion of an operon that governs growth on the sugar ribose. And there's no ribose in the environment, so there's no need to carry this around. And it happened that there's an insertion sequence near this operon, and it looks like very early on, another copy of that insertion sequence jumps into the ribose operon. Those two recombine and delete part of the operon. It doesn't provide a whole lot of a fitness benefit. I think it's about 0.5%, but it just happens really, really quickly.
Okay.
So that's an example of removing something that's unnecessary and getting a slight benefit from it. But there are other instances like the gene PYKF, which encodes an enzyme called pyruvate kinase, and it's just a part of the central metabolism of the cell. Every one of the populations has a mutation in PYKF, and none of them are ones that deactivate the gene. They just tweak its activity. We don't really know how. We have demonstrated that each of these mutations is beneficial. Each one confers about a 10% fitness benefit if we move that mutation into the ancestor. But we don't know the effect. We just know that these mutations are tweaking the activity of the gene. It's not introducing dysfunction.
Okay.
And then there are others, like there have been mutations in a number of the populations in genes like one called TOP-A for topoisomerase, and FIS, which I don't remember exactly what FIS stands for. But those two genes regulate how coiled the DNA is, and it relaxes the coiling. And this seems to change the topology of the DNA to affect gene regulation. And it alters the regulation of a bunch of genes. And just so I can give the listeners an image in their mind. So DNA is a very, very long molecule. Right.
It stretches out like a yard or maybe more. So then it begins to coil and then hypercoil and supercoil and keeps on getting all tangled up until you have this, scrunched up ball of DNA.
Exactly.
But there's no way that the enzymes can unpack the genes, the actual information, unless you tease apart some of those loops and knots. And that's what you're getting at here, that this mutation, there are two of them, I think you said,
right,
are just relaxing the DNA a little bit so some of the tangles can come out and enzymes can begin to read the information.
Right. We think that these mutations may change how tightly coiled DNA is in order to make it easier to activate some genes, we think.
Right.
And those are actually an example of a number of instances we've seen where a single mutation has an effect on a bunch of different genes.
Right.
So there are mutations in another gene called SPO-T and another called MAL-T, which all that you need to know about them is that they are what are called global regulators. So they regulate the activity of a slew of different genes. So these mutations mean that the activity of a huge number of genes throughout the genome in each of the 12 populations is slightly different. Now, do we know exactly how that's affecting fitness? No. We know if we put these mutations in SPO-T or MAL-T into the ancestor, it introduces a fitness benefit under the conditions of the experiment. But we don't know how, which is sort of a perennial problem that we have. It's easy to find mutations at this point. It's easy to study the fitness effects of mutation. It's really hard to figure out how the mutations are causing the changes in fitness, because that requires a lot of complex biochemical and molecular biology work. And
Okay.
yeah,
So the bottom line is you did see lots of changes, lots of genetic
right.
changes, and these are not, as the creationists and the ID proponents will often say, you weren't just breaking genes.
No.
Sometimes it's, so tell us about that.
Yeah, yeah. Like I said, like in the case of PYKF, there are a number of instances where it's clear that the mutation is introducing a change in the activity of the gene rather than turning it off entirely. So it's modulating the activity of the gene or expression of the gene. When we get in later in the conversation to talking about my work with the citrate users, there are actually a couple of different instances of this. So I'll talk more about that then. But yeah, we are not just seeing a matter of the breaking of genes.
Right.
And the thing is, yes, there are a lot of genes that have been deactivated. There are portions that have been deleted. But the thing that when intelligent design creationists speak of these, they speak of it as though there's something wrong with the breaking of these things. But the reality is, E. coli has this extensive toolkit of genes. Not all of them are needed under the particular conditions of this experiment. So there's no reason to keep them around.
Mm-hmm.
Ones that are expensive can be removed, or mutations are selected for that eliminate the function of the gene. And it's beneficial under these conditions. It's not that it's a bad thing. It's just
Right.
it helps with the adaptation to conditions. That is what natural selection does. It doesn't produce adaptation to everything. It isn't building Superman.
Right.
It's natural selection produces adaptation to particular conditions, and under these
Okay.
particular conditions, some genes aren't needed. So they get it's beneficial if they're broken by mutation.
So an analogy I like to use, because you said we're not trying to invent Superman here, let's talk about someone who takes up jogging and they get really good at it, to the point that they get it in their head that they're going to run a marathon. So they go through the training, they begin to change how they run, they get rid of some bad or energy wasteful things about their stride, they try to lose some fat. They'll start cutting out certain foods because they recognize that certain kinds of foods are just not good for an athlete. And they can actually change the way they view food. They can develop neuroses about eating certain kinds of foods. And they might decide to start ditching their watch and their glasses just to lighten their load. And they're going to start dressing differently. They're going to wear lighter clothes and running shoes. All of these changes involve losing or changing things in order to become a better marathon runner. And because of that, they can outcompete most people when it comes to marathon running. And maybe if the climate then changed towards a hot desert kind of climate, they might be better off and be able to compete better and outlast the overweight guy wearing the parka. But drop this marathon guy off in the middle of Alaska in the middle of November, despite all these marathon improvements in his light running outfit and fancy shoes, he's not going to compete very well. In fact, the overweight guy with the parka is probably going to do much better. Is that a helpful analogy?
Yeah, that's one way of thinking of it. Or another would be, if you look at the athletes in the Olympics, a pole vaulter is very unlikely to win the marathon. The marathoner is very unlikely to win pole vault because they trained a very particular them.
Right.
Yeah, there's this phrase that often comes up in evolution: the jack of all trades is the master of none.
Yeah.
Yeah, adaptation is the particular environmental conditions, and you don't need everything under a particular set of.
Okay, so we're not just breaking genes, and as I said, that is the thing that creationists and ID proponents will often say. You did mention citrate, and that's the one in particular they always focus on. So maybe, Zach, tell us a bit more about this whole citrate story again.
So as I noted earlier, the citrate's been the medium that we use in the experiment.
At this point, Zach and I got quite into the whole citrate story, quite a deep dive, and we're going to save that for next week. But Scott, I'd like to hear what you had to think about what Zach said.
Well, maybe the only comment I might say is that it was very fascinating. Maybe it's one of the first empirical study of evolution. In other words, not looking at fossils and paleontology and so on, which is empirical, but nonetheless it's historical. You know, you're trying to reconstruct something from the past, but this is in real time. You know,
right.
You're seeing evolution happening. Now, I know there have been other experiments along those lines as well, but like you said, 70 odd thousand generations, even though it's a blip on the evolutionary scale, it's certainly much more than we've ever done,
Yeah,
and to already find in even in that blip something happening. I
exactly.
mean,
Yeah,
right? It could easily have been that after 78,000 generations, and this would have been great fodder for the creationists, it could have been that absolutely nothing happened, nothing
right.
changed. And they might say, well, there you go,
Yeah,
even though it wouldn't have meant much because it is such a blip, right?
right,
So it really is, in a sense, kind of shocking that even in that little space of time, we do see things happening. And
right.
that bodes well for further research.
Yeah. And what makes it more surprising, yeah, I did ask this question in an interview, but I'm not sure the listeners really caught what was said. I asked about the stressors that were going on in this experiment, and Zach basically said, yeah, we weren't stressing them out. We were just giving them glucose and letting them compete against each other for the resources that were being provided to them. There was no stressors, no toxins, no penicillin, or nothing that they had to survive against. They were just simply having to outcompete each other. That's all they were doing.
Mm.
And like you said, Scott, they found lots of changes, even in that 75,000 generations.
Yeah.
So, Scott, I wanted to get your thoughts on a few other things that Zach said, and especially the fact that, completely contrary to what so many creationists and ID proponents will say, the LTE story is not simply about breaking genes, and it's not only about the citrate gene, but that's all that these anti-evolutionists ever seem to want to talk about.
Yeah. It demonstrates, I guess, that bias. I mean, they're hoping that the outcome of these experiments will be some definitive blow to the evolutionary model. And so I guess it's not surprising in a sense that they're going to attack the changes that they did observe and argue that they're not really a demonstration of evolution, right? And I suppose in some respects, that's okay. I mean, within the scientific context, you do have to scrutinize the results, right? And see, okay, is this really something that demonstrates the thing, or is it not? But I guess the frustrating thing is that once you do that, you move on.
Exactly.
But there
Yep.
tends to be a harping on, right, because of the bias there. And yeah, so that is unfortunate.
Right. One point that struck me while I was thinking about this after the fact, I'm not sure the listeners would have connected the dots on something that was said. I didn't connect the dots till I sat back and thought about it. Richard Lenski started this experiment with one single strain of E. coli. He put those into 12 different test tubes and then watched those 12 different populations of the original E. coli bug and how those evolved over time. And it occurred to me, this is just like how life began on Earth, from a single life form, and that single life form eventually made it to a test tube called Europe and Asia, and a test tube called the Americas, and another one called Antarctica, and another one called Australia. You get the idea. Half a dozen different geographical incubators, different regions, different climates, different opportunities. I guess that's where it's different from the experiment. They weren't different climates and things, but still, they were different evolutionary paths.
Mm-hmm.
And with that in mind, there's two really interesting points to make about evolution that we saw in this experiment. On the one hand, each of those earthly regions developed their own unique organisms. Only Australia produced kangaroos and wallabies and the platypus. Africa produced a giraffe and chimpanzees. Only the Arctic produced polar bears. All of these regions produced very different and unique organisms. And what we heard from Zach is that the bugs in these test tubes, they were different in some respects. But on the other hand, each of these geographically separated incubators produced organisms that look almost the same. Most of the world produced mammals that look like dogs. Australia produced a marsupial version of animals that looks like dogs, called the thylacine. If you saw the thylacine, you'd swear it's a genetic cousin of the dog. But marsupials and mammals are completely different lines of animals. They diverged and became genetically separate, like those 12 test tubes in different spots on the rack. They diverged more than 100 million years ago when dinosaurs ruled the world. The same thing happened with marsupial versions of cat-like animals, anteaters, the rhinoceros, squirrels, groundhogs, burrowing moles, the opossum, and deer. There are marsupial versions of these animals which you would swear are cousins of our North American ones, but they're completely different genetically. This is something called convergent evolution.
Right.
And so in the same way, Richard Lenski started the experiment off with 12 different test tubes, 12 different populations of the same original bug, which then began evolving off in 12 different directions. And between the 12 test tubes, they found 57 different genes that were changed to improve fitness. But many of the genes that changed were the same between all 12 test tubes, and many other gene changes were common between many of the 12 test tubes. Just like North America and Australia both produced a dog-like animal. So the 12 different populations got to the same endpoint, the same phenotype, but through different specific genetic mutations, just like North America did that through the mammal line, and Australia did that through the marsupial line.
That's really fascinating, because I assume the environment, the quote environment, and the environmental pressures on each of these test tubes were identical, right?
They were, yeah. That's where it's different from the experiment.
No, it is where it's different. But what's surprising about that is that given that, you would think that they would follow the same pathways, and yet what you're saying is that they kind of, like you said, they get to the same endpoint or similar endpoint, but they take different pathways to get there, and that's not due to environment. That must just be due to random mutation, right?
Right. Yeah.
Yeah.
Okay. Zach talked about that.
Yeah.
So I also want to unpack a bit more about those gene changes. Again, creationists like to simply say that the main findings of this Lenski study is about breaking genes. But what Lenski's group showed is pieces of genes were being shuffled around to make new combinations. And this is exactly what we find happening in nature. So, for example, Zach told us around the 16-minute mark that the distant ancestors of these E. coli were once able to grow aerobically on citrate in the distant past. But they lost that ability millions of years ago, and Zach said probably because they didn't encounter citrate under aerobic conditions in their ancient environment. Nature had found it useful to get rid of that function. But in the long-term evolution experiment, the bugs regained that ability to grow on citrate under aerobic conditions. And it wasn't through breaking any gene, but through insertion of genetic information into the citrate operon. And we'll hear more about that next week. And Zach gave other examples of information from one part of the genome being copied and pasted into new parts of the genome, leading to new control pathways and new ways to respond to new environmental conditions. So it's not about breaking genes, but about shuffling genetic information. And Zach referred to the fact that these cells have an extensive genetic toolkit, a toolbox full of not just whole genes, but parts of genes that can be moved around, on-off switches, or stretches of a gene that code for a unique and functional part of an enzyme, and all kinds of enzymatic tools to swap these pieces of information in and out all over the place. Anyone who thinks that that's a dangerous or unnatural thing for a cell to be able to do knows nothing about our immune system, because that's the whole reason we're able to respond to bacterial and viral threats so quickly and effectively. And by the way, I think it's also worth pointing out that in the same way that nature found it useful to have the E. coli bugs lose the ability to metabolize citrate under aerobic conditions millions of years ago, again, because the cells didn't encounter citrate in their environment under those conditions, here in the long-term evolution experiment, one of the 57 genetic changes they noticed was that the cells lost the ability to grow on ribose, because ribose wasn't in the solutions they were growing their bugs in. It's the same thing. We've heard the popular expression, life imitating art. Well, this is experiment imitating life.
Yeah, wow. That's a great way to put it. That's really, really interesting. You know, as you're talking, Luke, I think, and I know this is all analogies break down, but just for my simplistic mind, if we have 4 or 5 identical sets of cards, decks of cards, let's say 52 card decks, and you and I take those decks, and each of us, or even if one person, doesn't really matter, does a riffle shuffle on all 5 decks, and then we go through those decks, guess what? None of them are going to be the same, right? Now, and in some sense, I know it's a rough analogy, but it's that shuffling that's kind of going on. Is that right?
Yes, exactly.
Okay. Yeah. Okay.
Exactly.
So that's kind of how I can visualize it, I guess.
And that's good, because that's what I'm trying to get everyone to catch. It's not about breaking genes. It's about shuffling information.
Yeah, shuffling information. Yeah.
Yeah. Now, another thing that I want to point out, when Zach was mentioning some of the 57 gene changes that were identified in this study, he sometimes would say that this would give an advantage of 1 or 2%, or even in one case, 0.5%. And some listeners might think that advantages as small as that, those are just too small to be relevant. But I want to go back to the analogy I used with Zach about the guy training for the marathon. Think about all the things that an athlete will do to shave a few seconds off of a long-distance run. I mentioned the marathon runner ditching his watch and glasses and wearing very light clothing, very light running shoes. because these things shave off maybe a quarter of a pound off of his 150-pound weight. Think about the Olympic swimmers who will wear a latex cap on their head or even shave their head and maybe even their chest and legs to cut down on the frictional drag from their hair as they swim through the water. Or, for those listeners who do a lot of investing, maybe a financial analogy is better. Remember how much attention is paid to a stock that grows at 6% instead of 4%. That's just a 2% difference. Or a management fee of 3% versus 2.5%. Some investors get really squawky about that difference.
Yeah. I mean, I'm a fan of F1 racing, but it's interesting how in some cases, those races, man, the difference between between first and last place can be like a second,
Yes.
like it's, and yet that second is absolutely everything, right? It's the difference between being Max Verstappen, who's now on top of the world, and being somebody I can't even name because I don't know who they are. You know what I mean?
Exactly.
Yeah.
Okay. All right, Scott, one final point, one that's a little bit unpleasant to talk about. I made this point when it came to discussions around the bacterial flagellum, and here it needs to be said again about the long-term evolution experiment. Those anti-evolution people who have a reasonable scientific understanding, who are reasonably well-read in genetics, especially those with scientific letters behind their names, people like Michael Behe and Jonathan McClatchy and Stephen Meyer, they're fully able to comprehend that the LTE did not simply show breaking genes. Behe and his kind are fully able to comprehend that there were changes, some of which might have been destructive from a very narrow perspective, but just the same changes which built up new pathways and new genes. And to the extent that that's true, I have to accuse those people, Behe, McClatchy, Meyer, and their kind, who continue to misrepresent studies like this one. I accuse them of speaking in bad faith, intentionally distorting the truth. Deception, even.
Yeah, it's a real problem. This is the case where, in this area of science, I've seen this a number of years ago, and I've mentioned this on the podcast before. My youngest daughter, converted to Islam, and so I didn't know a lot about Islam, and I wanted to educate myself and come to an understanding and a acceptance of her new faith and so on. Doesn't mean I had to agree with it, but I wanted to understand it, right?
Yeah.
So I started to study a lot of material, both from I always go to the sources. So I'm reading the Quran, I'm studying Islamic scholars and so on. But I'm also searching for Christian kind of responses and so on. It was so disappointing to me to see so many, and this was particularly, I got to say, particularly amongst evangelicals, the gross misrepresentation of Islam, and it had to be a knowing misrepresentation because some of these were ex-Muslims.
Right.
There was a guy at Liberty University in particular who was the president. He was later demoted because of this, but he had written this book that somebody recommended. Oh, you got to read this book. This is the Christian response to Islam. I started reading it, and even with my little knowledge, I knew in the first few paragraphs that he was saying things that weren't true about Islam, right? Yeah. And so I looked into it. Well, the guy was a fraud. Turned out he was a fraud. And this book was super popular amongst evangelicals in the States. So in that area, I've seen exactly what you're talking about. Right. People that they know better, right? And I don't know if it's a zealousness, an enthusiasm that they feel like they're somehow promoting Christianity or something, but that's not the way to promote things.
Exactly. Yeah.
That if Christianity is anything, it's supposed to be about truth. Right?
Exactly. Yeah.
And so it just boggles my mind how often people can use untruth or lies to kind of promote the truth. And I'm not saying these guys are lying, don't get me wrong, but I totally get your frustration. And it's been really interesting for me. The privilege of being part of these conversations has been that a lot of these people that we've been talking, I really did look up to them at one point. And because they're so highly credentialed and because they could talk science, and I don't know science, it wasn't hard to convince me, right? But then when you hear them interacting or not interacting, with yourself, or if you bring other very competent scientists, you start to realize, oh, wow, really? So it's deeply disappointing.
It is. I agree. Yeah. Okay. I said it was an unpleasant topic. I put it on the table, and there you go. So let's move on from that. Next week, we're going to hear the other half of my conversation with Zach. We'll get into detail about that citrate story. So it's going to be another science-heavy talk. And we do that because, again, the creationists, the anti-evolutionists always want to focus on the citrate story. So we're going to hear in more detail. But Zach does do a great job bringing it down to a non-expert level, and then I bring it down a couple more notches, maybe to a high school level.
So tune in again and bring a couple friends while you're at it. And we'll see you next week.
Bye for now. You've been listening to Recovering Evangelicals with your hosts, Luke Jansen and Scott Dyer. Send your comments and suggestions for new episodes to lukejjansen@gmail.com. Join our private discussion group on Facebook by searching for the Recovering Evangelicals podcast group. Look for the logo that you always see at your favorite podcast catcher and ask to be let in. Subscribe to lukejjansen.wordpress.com if you want to get advanced releases and group posts, or browse through our archive for a full listing of all our previous episodes. And please help the podcast by sharing with a friend and leaving a review at your favorite podcast catcher.